So low temperature is also very stressful to the plant growth and development. So when the plants are exposed to low temperature, say for example, when they are exposed to 10 to 15 degrees centigrade of temperature, then it causes the chilling stress. When the temperature reaches below 0 degrees centigrade, it freezes the plant materials and it causes the freezing stress in plants. So the plants which are adapted to tropical and subtropical climates, when they are exposed to such kind of temperature, it leads to the stressful condition in them. Okay, so these tropical and subtropical climates are adapted to the warm temperature. When they are grown at these temperature, it becomes stressful to the plants. Okay, next. So these tropical and subtropical species are susceptible to chilling injury when the plants growing at a temperature of around 25 to 35 degrees centigrade, which is the optimum temperature of the tropical and subtropical plants, when they are cooled to around 10 to 15 degrees centigrade, chilling injury occurs. Now freezing injury occurs when the plants are exposed to the temperature below the freezing point of water. Okay, when it's in the minus, when the temperature is in minus, then it causes a freezing injury. What are the effects of freezing and chilling injury? We will see in some time. Now in some plants, the reproductive development is spatially very sensitive to chilling temperature. For example, when the rice plants are exposed to chilling temperature at the time of anthesis. Now what is anthesis? It is a floral opening. So when the rice plants are exposed to chilling temperature at the time of anthesis, it results in sterile flowers. Flowers are not fertile at that time when they are exposed to chilling temperature at the time of anthesis. Now some plants shows resistance to the chilling and freezing stress when they are acclimated. Acclimated means when they are made resistant by the exposure to the cool temperature again and again, then they become resistant. They become hardened or acclimated. They become resistant. We'll use the word resistant for acclimation also. So when they are exposed to the chilling temperature again and again, that means we are hardening it. These hardened plants shows acclimation by exposure to the cool and non-injurious temperature. But when there is a sudden exposure to near 0 degrees centigrade, then it causes a cold shock in plant. The sudden exposure to such a low temperature causes a cold shock in plant, and this causes the injury in the plant. The chances of the injury increases when there is a sudden exposure. Okay. Next, the effect of cold stress. So at first, when there is a chilling or freezing injury, what happens? The membrane properties change. And when there is freezing injury, the ice crystals are formed and protoplast is dehydrated. Okay, so this ice crystal formation is mainly during the freezing injury. And when there is chilling, now the effect of cold stress, when the plants are exposed to this low temperature, then their membrane properties change and the ice crystal formation and protoplast dehydration occurs. Okay, so we see that when the plants are exposed to low temperature, say around 10 to 15 degrees centigrade, then membrane properties are changed. And when the plants are exposed to below 0 degrees centigrade, which is the freezing temperature, which causes the freezing stress, it causes the ice crystal formation and protoplast dehydration. So under 10 to 15 degrees centigrade, there will be chilling injury, which causes the change in the membrane property, and when the temperature is below 0 degrees centigrade, it causes freezing injury, which leads to ice crystal formation and protoplast dehydration. So what happens when the membrane properties are altered? Let's see. So when the leaves are exposed to chilling stress, chilling injury occurs. So during chilling injury, the plant shows inhibition of photosynthesis. Carbohydrate translocation rate is slowed down. Respiration is respiration rate is lowered. Protein synthesis is inhibited and degradation of existing proteins. So all these responses, they depend on a common primary mechanism which involves the loss of membrane function. So when the membrane function is lost, these many pathways are inhibited. Or better I write, these pathways are affected. Okay. The physical properties of the lipids, they greatly influence what they influence. They influence the activity of integral membrane proteins. For example, they influence H plus ATPase. So H plus ATPase is an integral membrane protein which is inhibited in response to chilling stress. They also inhibit several carrier proteins and channel forming proteins. So these carrier and channel forming proteins, which used to transport the ions and solutes, they all are affected. Also, when the chilling sensitive leaves are exposed to high photon fluxes and chilling temperature, they are photoinhibited. So we saw in drought stress and as well as in high temperature stress that the plant shows photoinhibition. Now the effect of cold stress, when the plants are exposed to this low temperature, then their membrane properties change and the ice crystal formation and protoplast dehydration occurs. Okay, so we see that when the plants are exposed to low temperatures, say around 10 to 15 degrees centigrade, then membrane properties are changed. And when the plants are exposed to below 0 degrees centigrade, which is the freezing temperature, which causes the freezing stress, it causes the ice crystal formation and protoplast dehydration. So under 10 to 15 degrees centigrade, there will be chilling injury, which causes the change in the membrane property, and when the temperature is below 0 degrees centigrade, it causes freezing injury, which leads to ice crystal formation and protoplast dehydration. So what happens when the membrane properties are altered? Let's see. So when the leaves are exposed to chilling stress, chilling injury occurs. So during chilling injury, the plant shows inhibition of photosynthesis. Carbohydrate translocation rate is slowed down. Respiration rate is lowered. Protein synthesis is inhibited and degradation of existing proteins. So all these responses, they depend on a common primary mechanism which involves the loss of membrane function. So when the membrane function is lost, these many pathways are inhibited. Or better I write, these pathways are affected. Now the effect of cold stress, when the plants are exposed to this low temperature, then their membrane properties change and the ice crystal formation and protoplast dehydration occurs. Okay, so we see that when the plants are exposed to this low temperature, then their membrane properties are changed and the ice crystal formation and protoplast dehydration occurs. So all these responses, they depend on a common primary mechanism which involves the loss of membrane function. So when the membrane function is lost, these many pathways are inhibited. Or better I write, these pathways are affected. Now the effect of cold stress, when the plants are exposed to this low temperature, then their membrane properties change and the ice crystal formation and protoplast dehydration occurs. Okay, so we see that when the plants are exposed to low temperatures, say around 10 to 15 degrees centigrade, then membrane properties are changed, and when the plants are exposed to below 0 degrees centigrade, which is the freezing temperature, which causes the freezing stress, it causes the ice crystal formation and protoplast dehydration. So under 10 to 15 degrees centigrade, there will be chilling injury, which causes the change in the membrane property, and when the temperature is below 0 degrees centigrade, it causes freezing injury, which leads to ice crystal formation and protoplast dehydration. So what happens when the membrane properties are altered? Let's see. So when the leaves are exposed to chilling stress, chilling injury occurs. So during chilling injury, the plant shows inhibition of photosynthesis. Carbohydrate translocation rate is slowed down. Respiration rate is lowered. Protein synthesis is inhibited and degradation of existing proteins. So all these responses, they depend on a common primary mechanism which involves the loss of membrane function. So when the membrane function is lost, these many pathways are inhibited. Or better I write, these pathways are affected. There are some bacteria which also help in the ice nucleation. Okay. Say for example, the bacteria Pseudomonas syringae and Erwinia herbicola, they are the two kinds of bacteria that have a kind of protein on their surface. These proteins are known as ice nucleation proteins. These proteins are known as ice nucleation proteins. So these proteins aggregate the water molecules and helps in the formation of ice crystals. Okay, they helps in the formation of ice crystals. And these bacteria, they are found mainly when the leaves are cooled to a temperature of -3 to -5 degrees centigrade. Okay, -3 to -5 degrees centigrade. Now, so they naturally inhabit the leaf surface, and these kind of bacteria are mainly found in the region where the snow is present. Like in the freezing temperature, these bacteria are found. Now, in response to cold stress, the plant shows several kinds of mechanisms. The numerous genes are induced during cold acclimation, and for freezing tolerance, they synthesize antifreeze protein, in short we will write as AFP. They synthesize osmolytes, proteins for membrane stabilization, and Lea proteins. Now, we saw that Lea protein was synthesized in the drought stress also. Okay, so this proteins also helps to combat with various stress response. Now, limitation of ice formation. When they limit the ice formation, they can tolerate this freezing temperature. Okay, so see, as long as the freezing of water is confined to the apoplast, so freezing can be inside the cell or it can be outside the cell. So before coming to this slide, I will explain that freezing can be intracellularly inside the cell, or it can be extracellularly, which is outside the cell. Now, I discussed many a times extracellularly means outside the cell, that is in the apoplastic region. So when the ice formation is in the extracellular region, so the physical properties of the lipids, it greatly influence the activity of channels and carrier proteins. So these channel and carrier proteins, they regulate the transport of ions and other solutes, as we all know. So one of the protein which is inhibited is H plus ATPase. Okay, so the activity of integral membrane proteins, for example H plus ATPase, carriers and channel forming proteins, they are highly regulated and affected by chilling stress. So again, when the chilling sensitive leaves, when they are exposed to high photon fluxes, the plants shows photoinhibition. And once the plant shows photoinhibition, their photosynthetic machinery are damaged. We already studied this in light stress as well as water stress and high temperature stress. So look here, when the chilling sensitive plants, when they are exposed to chilling temperature, when they are exposed to chilling temperature, very low temperature, and when they are exposed to high photon fluxes, then what we see, that they shows photoinhibition. Okay, and once photoinhibition occurs, this results in the damage to the photosynthetic machinery. Okay, so what we studied, that H plus ATPase is a protein which is a transport protein which is hindered in response to chilling stress. Also, when to the chilling sensitive plants, when they are exposed to chilling temperature which is around 10 to 15 degrees centigrade, and when they are exposed to high photon fluxes, it shows photoinhibition, and photoinhibition leads to the damage to the photosynthetic machinery. Okay. Now, very important term, transition temperature. What is transition temperature? It is the temperature at which the membrane changes from the fluid to the semi-crystalline state. Okay, so there are two conditions. The conditions when the transition temperature, so it favors the formation of semi-crystalline state. So to form the semi-crystalline state, that means the transition temperature increases. Again, back to form the fluid state, the transition temperature decreases. Okay, to form the semi-crystalline state, transition temperature increases, and to form the fluid state, the transition temperature decreases. Now, what happens in chilling sensitive plants? The sensitive plants are prone to form the semi-crystalline state of the membrane. So they are favoring this semi-crystalline state formation. So that means transition temperature is higher in sensitive plants. Clear? They have higher transition temperature. Okay, we all know that the chilling sensitive plants have more saturated fatty acid. And chilling resistant plants, that means they are able to keep the membrane state in the liquid phase. So they can favor they favor the fluid state. That means transition temperature here is lower. Okay, and in the chilling resistant plant, there is lower proportion of saturated fatty acid and there is higher proportion of unsaturated fatty acid. Clear? Now this concept is very important. Lot of question comes based on this. Okay. So what happens in the acclimated plants? So I already told these acclimated plants are the resistant plants. These resistant plants will also have the same characteristics as the acclimated plants. Okay, so acclimated plants also have this lower transition temperature and high proportion of unsaturated fatty acid and high proportion of unsaturated fatty acid. Okay. So one question which came in December 2013, the membrane of chilling sensitive plants are characterized by. So at first, let's take the wrong ones. Higher proportion of saturated. So according to our conception, chilling sensitive plant have higher saturated fatty acid. And what is about their transition temperature? This chilling sensitive plant favors the formation of fluid to semi-crystalline. So when they favors fluid to semi-crystalline, in that case transition temperature is higher. We discussed in the last slide. So now let's solve the question. So they have higher proportion of saturated fatty acid, correct? Lower transition temperature, wrong. Lower transition temperature, again wrong. I'm not reading further. Lower proportion of saturated fatty acid. So can you find that A is the correct option? Now coming to the ice crystal formation and protoplast dehydration. So this is a response to freezing injury, which is below 0 degrees centigrade. Okay, this varies greatly among tissues. The ability to tolerate freezing temperature varies greatly among tissues. Now seeds which are under dehydrated conditions and fungal spores which are they are not in the active condition, so they can survive the high temperature range. So they can survive, some can survive near absolute zero. So indicating that these very low temperatures are not very harmful to some of the seeds and other dehydrated tissues. So ice usually forms within the, so they are formed first in the intercellular spaces and in the xylem vessels from where they quickly propagate. So in the two regions they first form, that is the intercellular space that is in between the cells, okay, in this region and in the xylem vessels from where they quickly propagate. Now this ice formation is not lethal to the hardy plants. I already mentioned many times that hardy plants are the acclimated plants or the resistant plants. So these hardy plants can recover when they are fully warmed. Okay, when they are in the warm condition back. Now when the plants are exposed to freezing temperature for an extended period, then the growth of extracellular ice crystals result in the movement of liquid water from the protoplast to the extracellular ice, causing excessive dehydration. So when they are exposed to the extended period, see, so these are when the cells, okay, this is inside the cell where the protoplasts are present, and here first when the ice formation starts, ice crystal formation, when it starts here, when the plants are exposed to the freezing temperature for a longer period, then what happens, this ice crystals results in the movement of liquid water from the protoplast to the extracellular region. Okay, this leads to the excessive dehydration. So all the water comes out from the cell, causing excessive dehydration when they are exposed to the longer extended period. Now we see that to form the ice, several hundred molecules are needed to form the ice crystal. So the process whereby these hundreds of water molecules starts to form a stable ice crystal, this is known as ice nucleation. Okay, where many molecules unite together to form a stable ice crystal, this is known as ice nucleation. Okay, there are some polysaccharide and some proteins which favors the formation of ice nucleator. Some ice nucleator are also made by bacteria. Okay, what do they do? These bacteria help in the aligning water molecules along repeated amino acid domains. So what we said, that this ice crystal formation can be inside the cell and outside the cell. So once the ice crystals are formed outside the cell, they may not cause immediate physical damage to the cell. So these ice crystals are formed at first extracellularly, then intracellularly. Okay, when they are formed in the extracellular region, what happens? That they cause cellular dehydration. Now, how they cause cellular dehydration? So see, here in the extracellular region, once the ice crystals are formed, what happens? They are combining the water molecules. The free water molecules are not available in this region anymore. So because of which what happens? The solute concentration in the extracellular region increases. Because the water molecules became less, solute concentration increases. Now when solute concentration increases, that means that water potential, the sign of water potential is psi W. So when solute concentration increases, the water potential decreases. So this region is extracellular region, and inside the cell is intracellular region. So what we saw, that the free water is not available, it causing the water potential to become negative. So we always know that water moves from higher water potential to lower water potential. So outside the cell, water potential has lowered down. So from inside the cell, the water will come out. From higher water potential to the lower water potential. Because ice has first formed in extracellular region, it will later form in the intracellular region. So once this extracellular region, the ice has formed, it will help in the, or it will cause the release of water from the cell. So this leads to protoplast dehydration. Here. So inside the cell we can say it's a symplastic pathway, and outside the cell it's an apoplastic pathway of movement. Okay. So here what pathway we observe, that there is a movement of water from the symplastic pathway to the apoplastic pathway. Clear? Now, there are some cells which we already discussed, like seeds and pollens. They are very less affected by ice crystal formation. Okay. Now, in response to water stress, one more thing, that this intracellular region, when the ice once formed in the intracellular region, then we say that it has adverse effect on the plant. Okay, it shows, we will see in few slides, it has a lethal effect on the plants. Now, ethylene, in response to cold stress, ethylene becomes activated. It is synthesized more and more, and it induces the transcription of proteins named AFP proteins or antifreeze proteins. So ethylenes are activated in response to cold stress, and they help in the synthesis of AFP, which are antifreeze proteins. Now, once antifreeze proteins are synthesized, they will be released through endoplasmic reticulum, Golgi bodies, vesicles, which will fuse with the plasma membrane, and they are then deposited on the surface of the cell wall. Okay, so these antifreeze protein, once they are released, they will fuse with the surface of the cell wall, and they will release the antifreeze proteins on the membrane and helps to inhibit the ice crystal formation. Okay, so these are the vesicles which release AFPs. Okay. Now I told that some osmolytes will also be synthesized in response to stress, this low temperature stress. That sugars and some of the cold inducing proteins are synthesized in this low temperature stress to combat with the stress, and these sugars and some cold induced proteins have cryoprotective effects. What is the meaning of cryoprotective effects? That they are able to stabilize the proteins and membranes during the dehydration induced by low temperatures. So sugars and some cold induced proteins, they are able. Now, glycoproteins accumulate and the protoplasm becomes generally more resistant to dehydration. They accumulate a protein named glycoproteins. In winter, it was found that when the sucrose concentration was increased, the plant shows freezing tolerance. So, sucrose concentration is helping in the freezing tolerance. Now, ABA is a stress hormone. It is produced in various kinds of stress response. So, in cold stress, also ABA is synthesized. So it was examined in the mutant of Arabidopsis, that when there is a mutant in ABA, when the Arabidopsis are insensitive to ABA, the mutant of Arabidopsis which are insensitive to ABA, or or Arabidopsis which are ABA deficient. Okay, so both the mutant and the Arabidopsis deficient so both the mutant of ABA and the ABA deficient Arabidopsis, they are unable to show low temperature acclimation, because ABA is an important hormone that also helps to combat with the freezing stress. Okay, so when ABA was not there, it was seen that they are not able to acclimate to freezing. Now, numerous genes are induced during cold acclimation. Now, during heat stress, we studied that heat shock proteins are synthesized. So, heat shock is a new kind of protein which will be synthesized in response to heat stress. But uh, the housekeeping genes, the genes which are always active, are shut down. So, in heat stress, what we saw, that the normal genes, the housekeeping genes which are always on, they are shut down, and the new genes are synthesized. But in cold stress, none of the genes will be down regulated. Okay, so in cold stress we see that the housekeeping genes are not down regulated. Instead, some new genes are transcribed and translated, but the housekeeping genes were unaffected in cold stress. So in cold stress, the housekeeping genes are not down regulated like in heat stress. Okay. So, in response to freezing stress, the plant synthesized a kind of protein named as antifreeze protein, which was discovered first in the cold acclimated winter hardy monocots. Now, it was seen that these antifreeze proteins belong to a class of protein named as endokinases and endoglucanases. So these endokinases and glucanases, they are produced in response to different pathogens. These two classes of proteins, they are produced to fight with the pathogens. Okay, these proteins are grouped under pathogenesis related protein known as PR proteins. Okay, so in response to pathogens, some PR proteins are activated. These PR proteins are the class of endokinases and endoglucanases, which helps the plant to fight with the pathogen. Defense to pathogen attack. Okay, so here what we see that these antifreeze proteins, since it has a role of endokinases and endoglucanases, they help the plant to fight with the pathogens also. Okay, so they protect the plant against both cold stress and pathogen attack. So this is very interesting to know. Now another class of protein which are induced in response to cold stress are LEA proteins, which are like produced in response to ABA. So ABA helps in the formation of LEA. Okay, in response to ABA, LEA proteins are formed, which are synthesized which are also synthesized during the dehydration stress. Now there were two proteins named COR15A and WSE120. They interact with the membrane, which enhances their stability to low temperature as well as against freezing. Now, because the formation of extracellular ice crystals, it generates severe osmotic stress. Now, why osmotic stress? Because it helps the cells to lose water. Because the ice crystals are formed outside the cells, the water is reduced outside the cell. So once water is reduced, that means the solute in that region is increased. So when water is reduced and solute is increased, it causes the negative water potential. So whenever the water potential is negative, then what we see that the water will move to the region where the water potential will be negative. So here if it's negative, then here it is positive. Then water always moves from positive to the negative water potential. Water will come out of the cell, which leads to the dehydration of the cell. Okay, so what we see that because the formation of extracellular ice crystal generates a significant osmotic stress also. Now this is known as osmotic stress. Okay, due to solutes, when the stress is increased, it's osmotic stress. Now coping with freezing stress also requires they have to cope with the osmotic stress. Now how they will cope with the osmotic stress? Now it is found that so how the plant will cope with the osmotic stress, this we have already discussed while we were doing the water stress response. Okay. Now, it was seen that in cold response, the promoter regions of certain cold regulated genes are activated. So when there is dehydration stress or low temperature stress, the promoter region of certain cold regulated genes are activated. So this has led to the identification of a kind of element named DNA regulatory element. Okay, so what I meant that in response to cold stress or dehydration stress, the promoter region of certain gene is activated, which has led to the identification of an element named as DNA regulatory element. Sorry, dehydration responsive element, which has led to the identification of DRE, dehydration responsive element. Now, this uh, dehydration responsive element have a specific sequence named CCGAC. Okay, CCGAC. This is dehydration responsive element. which is present in the promoter region of certain cold regulated genes. And these DRE are activated in response to low temperature and dehydration. Now there are some specific proteins which will bind to this DRE element. They are known as C-repeat binding factor or CBF. So with DRE, CBF proteins will bind. So these CBF are transcriptional activators that are involved in regulating the expression of cold regulated genes. So this is a cold regulated gene. Okay. So the binding of CBF1 and DREB1 B, it is very unique in response to cold stress. So this unique combination is seen in cold stress. Okay. Now, osmotic and salinity stress also activate this, also facilitate the binding of CBF and DREB, but the number is different. Okay. So see, the DRE binding element of the DREB2 type are induced only by osmotic or salinity stress. So by cold stress, it is CBF1 and DREB1B, DRE-DREB1B in response to cold, and in response to, whereas DREB B2 is active only in the osmotic and salinity. Okay, just remember 1B is specifically for cold. Okay, this combination helps in the cold acclimation. So what we studied in summary, that the cold stress causes the inhibition of several transport proteins. Okay, they inhibit H plus ATPase. So when H plus ATPase is inhibited, several pathways will be inhibited. So to help the metabolism to go on, plants synthesize, in plants, H plus pyrophosphate or pyrophosphatase work. They work when this H plus ATPase are not working. So they helps in the proton. They will help to create the proton gradient in a situation when the ATP supply is depleted. Okay. So because of chilling, when the ATP supply is depleted, when H plus ATPase cannot work, H plus pyrophosphatase works. It will be pyrophosphatase. Okay, it was also found that to combat with the stress, ethylene was biosynthesized. Ethylene is when biosynthesized, it helps in the formation of, this works, it helps in the formation of transcription and translation of antifreeze proteins. Okay. This increase in ethylene also causes several response, like it helps in leaf abscission, it helps in senescence. Okay, it helps in wound healing if there is any pathogen attack together with cold stress, increase disease resistant. Okay, so these are all responses showed by the ethylene, and ethylene also helps in the synthesis of AFP, which are antifreeze proteins. So these all together helps in the plant to acclimate with the cold stress. So a question will solve. Following are some statements about low temperature stress in plants. Fatty acid composition of mitochondria isolated from chilling resistant and chilling sensitive plant differs significantly. So resistant plant and sensitive plant will have different composition of fatty acid. Okay, one will have unsaturated and another will have saturated. Okay, ratio of unsaturated fatty acid to saturated fatty acid is lower in chilling resistant plant. No, in chilling resistant plants, unsaturated fatty acid will be more. So this is a wrong statement. The cellular water does not freeze at even minus 40 degrees centigrade because of the presence of solutes and other antifreeze proteins. The solutes helps the plant to prevent the osmotic stress. And antifreeze proteins help the plant to prevent crystal formation. Yes. Should prevent the formation of ice crystal. Okay. Now heat shock so this is correct. Heat shock proteins do not play any role during low temperature stress. So I told there are some heat shock proteins which are produced in response to heat stress only, and there are some proteins which are produced in various kinds of stress. Okay. So it's not that they are only produced in response to heat stress. They can be produced in various kind of stress response. So this is wrong. So A and B. So which one are correct? We saw that P and R are the correct ones. So B is the correct option.